talk this year by a UC Merced professor. Actually, the first two weren't professors, but tonight she's in the, I forget the name of it. It's got a long name for the department, excuse me. We'll get there. The Department of Life and Environmental Sciences, they have a great department. So if you're interested in studying the environment, go to UC Merced. You'll see what kind of great professors they have. I just want to read one thing. I'm not going to go through all this stuff. You can see she got her PhD from Santa Cruz, her BA from Dartmouth. She's also went to Chicago. But let's look at what her boss said from UC Merced, the lower left. Sora is a great asset to our department, school, and broader campus. Not only is she a distinguished early career scientist and engaging teacher, she's also a phenomenal leader, innovator, and mentor in equity, diversity, and Yay.
Still am. Not back down.
As a department chair, I value her focus on people and community building efforts, and that I can always rely on her for great ideas. Please welcome Dr. Sora Kim.
We still have people standing in the back. Raise your hand if you have seats. Please, it's going to be a 45-minute to an hour talk, so come sit down. Yep, there's 2 seats there and there.
Yes. Oh, is this too loud? It's always weird to hear my own voice. Okay? All right. Cool. This is a huge crowd. I'm kind of nervous. So my name is Sora Kim. I'm a professor at UC Merced in the Life and Environmental Sciences Department. And I'm here to talk to you about Beyond Shark Week, environmental change based on chemistry and fossils. And so I'm actually not trained as a biologist, I'm trained as a geologist. All of my degrees are from geology or earth sciences and environmental science departments, but I am sort of an interloper, and it turns out people really like sharks. I'm always amazed at how much people like sharks. So just some nice images. These first two here. This one is a cover of one of the journal articles that I recently published, and then this one, it may have also been a front of a journal, but I have a good friend. She actually worked for me when she was an undergrad, helping me take care of sharks in captivity. Now she's a scientific illustrator at the Smithsonian Natural History Museum, and I got to see her last week, so it's cool to like keep working like that science and art continuum with a former undergraduate student. And then on the far side here, this is from Ray Troll, who's a really famous artist and also a paleontologist that does a lot of great work. And he's actually given me permission to be able to use all his artwork throughout my various talks, which I really appreciate. So, how many of you have watched Shark Week? A lot, but not everyone. Okay, I had actually never watched Shark Week until I was on Shark Week, and then I was like, I guess I should watch this. So I was on this show called Jaws versus Meg, and then last year I also did another session with Jaws versus the Leviathan, and so that's an image there. And for those of you that watch Shark Week, you might notice that there's a theme in Shark Week, and it is white sharks. And it's like, white sharks, ride or die. But that's not the only species of sharks. There are actually almost 500 living species of sharks, skates, and rays, which is a lot of diversity. And this image is a phylogeny of all of the sharks, skates, and rays, the chondrichthyans. And it is showing the amount of molecular data coverage that we have. So genetics is like a really popular tool that we have in science. And where it is blue is not very well known, And then where it is red means that we have 100% coverage. And so where you see the red bars here, this is for laminiforms, is where white sharks, salmon sharks, mako sharks, thresher sharks, where those all sit. And then this other area that is orange is the echinorindiforms, and that's because there's only 2 species. So it's like a lot easier, you're gonna get total data coverage if you have 2 species. So this is the state of kind of like a shark knowledge, honestly. And being that I came into this from geology, I work on this method. It's called stable isotope analysis, which you all will hear a lot about tonight. And I was like, well, you know, I'm interested in sharks in the fossil record, but you know, the biologists, they have it all figured out, so I'll just like pull from what they know and then apply it to, you know, the ancient sharks. And then I was like, lo and behold, they actually know very little. So I have had the great fortune to be able to sort of make that my career in bridging between modern and paleoecology and learning about sharks. I do everything from like physiology to modern sharks, like where they're moving, what they're eating, to ancient sharks and sort of how past climate change may have impacted them and how they survived. It was really like my inner like happy place to think about. So I want to start off with some, well, there's going to be a number of questions here. And there will be some prizes from UC Merced, as I am representing here. What shark is found in the Mississippi River? Anybody know?
Bull shark.
Wait, who called that out?
Bull shark.
Yes, bull sharks. You can come up afterwards and get a prize. Bull sharks, that's right. This is what they look like. They are fairly ferocious looking. They will really eat anything. But they are actually in the IUCN, which is the International Union of Concerned, what is N? I always forget what N is. Well, it's this red list that a lot of scientists have put a lot of time into trying to learn about a particular species, but it is listed as vulnerable. And so this is the distribution of bull sharks. They're actually quite widely spread throughout the world, but in nearshore areas. But the place that a bull shark has been found is in St. Louis up the Mississippi River. So one thing that I have realized is that I'm from the South. I'm from Georgia, actually. And when I moved to California, I was like really surprised that people like their sense of geography kind of ended at Nevada. So that's what I always got. I wanted to put a star up here for everyone. So that we do continue on past Nevada and St. Louis. It's in the middle of the country. It's like really far inland. That is not where you would expect to see a shark if you were swimming in the water. You know. All right. So some common ways to study sharks is observation. So this is an image of someone diving with a whale shark, and I mean, whale sharks, they're not apex predators necessarily, but they are big. They're actually quite scary to see in the water. It is probably the shark that I've been the closest to, but that has also scared me the most. Despite, like, I've been in a cage with a white shark, but a whale shark scared me way more because they're just so huge, and to be next to something so large is incredible. But there are other shark species that you do not want to be in the water with without a cage or some sort of protection device. The other way that we study sharks, especially what they eat, are stomach-gut contents. For those, here they have like a really small shark, and sharks can invert their stomachs, and that's like sort of a defense mechanism almost for them. And so they're just pulling out the stomach and then emptying out its contents. But usually how scientists look at stomach-gut contents is that they actually sacrifice the shark, which let's kill the shark, and then empty out its stomach contents. So that's not really a great way to go about conservation, to have to like take out the stomach and count up all the bits. Another way is to do tagging. This is an image from this tagging of pelagic predators. And they put, this is from satellite tags that they put on sharks. And they do it on whales and turtles and birds and all kinds of animals. But it's actually a satellite tag that tracks the animal. their goal is to get like 3 years of data, but oftentimes the tag pops off early. But each one of these tags is somewhere like $10,000 to $15,000. So there's only so many animals that you can tag when the price tag is that big. And then lastly, this method that I sort of started on is called stable isotope analysis. And so this is a method that's actually was commonly used in geology, but has now started to be used more and more in ecology. And it is one of the primary ways that we actually study sharks now. And so that's kind of exciting to be like, yes, I helped start this. And so to tell you a little bit about stable isotopes, it's probably something that you may have learned in high school chemistry, and maybe there's some cobwebs there, so I wanted to just go ahead and clear it up. Stable isotopes are the same atom, but there's different number of neutrons. So to remember, this is carbon 12, this is carbon 13. So in the nucleus, there are protons and neutrons. The number of protons is what determines what element it is, but there can be different numbers of neutrons, and there are just small differences in weight. They don't actually change the charge of the element or the atom, but there's just these really small parts of weight, differences in weight, and they're usually like less than 1% of all the carbon atoms on Earth basically are this carbon 13. There's also carbon 14, but it's unstable. That's what we use to do radioactive decay. There's radioactive decay and C14 dating. It's where how we can get the age of some younger fossils. But carbon 12 and carbon 13 are totally stable, totally safe. Like all of us have carbon 13 and carbon 12 in us. Carbon isotopes out, in nature, they actually tell us something about how energy is flowing through an ecosystem. So here I have a chlorophyll concentration map of the northeastern Pacific Ocean, and you can sort of see that where chlorophyll is the highest is in red, where some of those places might be with upwelling zones along the California coast, and then where chlorophyll concentration is actually quite low in tropical warm waters. And so there are these gradients of carbon 13 abundance and also nitrogen 14 versus 15 abundance. There also this other aspect of isotopes, like how I told you that there's some small differences in weight, that they change and they behave differently through chemical, physical, and biological processes. And one of those is through your diet. So all of the nitrogen that you acquire is through mostly the protein that you eat. And that's why it's important that even like vegetarians, and especially vegans, you have to watch, make sure that you make your protein content. Like if you're working out and you want to build muscle, you have to make sure that you have more protein. And that's because all of that is going into your nitrogen. It turns out that we also produce waste. So when we breathe, we respire CO2. That carbon that we're respiring, it has more of the carbon 12, and our bodies keep the carbon 13. And then in terms of nitrogen, we pee and poop, and our waste products have more of the nitrogen 14, and then our bodies retain this nitrogen 15. So there's this trophic enrichment that the nitrogen 15 isotopes, they actually sort of biomagnify as you go up the food chain. So those are sort of how we use them in modern ecological studies. Now I'm going to ask the question of what do white sharks eat? They do eat fish. What else?
Seals.
Tuna. Seals. Yes. So these are all correct, actually. So white sharks have this incredibly varied diet. Oftentimes people only give me the answer of seals and sea lions. So good job that you all actually know that white sharks eat other things other than seal babies. This is what we like to show on Shark Week because it's like, I don't know, pulls at the heartstrings. But actually there have been, accounts of sharks eating birds. That does not seem like very nutritious, like a big meal though. And then lots of fish as well. And then there's also been squids in its stomach. So we actually, from looking at the isotopes that I told you about, and probably probably like more detailed than you've ever thought about isotopes, that white sharks actually, they eat this huge, diverse array of diets. And we see that in their carbon and nitrogen isotope composition. So here, I'm actually showing carbon isotope variation along the x-axis and nitrogen isotope variation along the y-axis. So you can almost think about nitrogen like trophic level, and then carbon, sort of like this would be more near shore, this would be more offshore oftentimes. And then each color is a different shark. And I actually got this data from white shark vertebrae, and after the talk you all can come and like look at these different specimens. This is a vertebrae. And I drilled out every single band and got this data. And so this is all the prey that I didn't sample these, I got these from scientific literature, and I just labeled them for you to get a sense of just how variable their diets are. Like, there's a lot of stuff up there. And including like, this individual, it just looks like it's hanging out like between tuna and squid. Cephalopods are squid. So it is like basically a sushi dinner right there. And then we also have things like sea otters, other sharks, and then here's our marine mammals. And one of the questions, the reason why I started this project was actually that I was curious, did white shark diet change as they got older? And so, you've probably heard the song Baby Shark, but Baby Shark is small, and then as sharks get bigger, they actually get bigger mouths, and so they can actually eat bigger things. And so that was always thought that like sharks, they had this shift in diet once they got large enough. And when I looked at the nitrogen isotopes in these annual bands of their vertebrae, they're like tree rings basically that you can like count up in, the bands, but you can also drill them out and then analyze their isotopes. I found these different patterns. So the first pattern that I saw was that there is a shift when the white sharks are about 6 years old. So that's when they're sort of like this juvenile state. The second trend that I saw was that there's a gradual increase with age, that they're like sort of steadily increasing. And then the last pattern that I saw was that there's absolutely no pattern, that it's totally stochastic. Some of them bounce around all over the place, like the one at the top here, this one here, and then WH128. But then there's others that are like pretty steady. This CC3, this was a white shark that they brought to UCSC and dissected. I think it was like in 2003. It was before I was there. But that's one of the more recent sharks, and it actually has a very flat diet. And one of the things that I actually thought going in was that there's this thing called the Marine Mammal Protection Act, which made it illegal for fishermen to kill like seals and sea lions, and that was a way that these seal populations really rebounded, especially in California. So we see this huge increase in the seals and sea lions, but we don't actually see their diets, the white shark diets actually changing as a result of it. So that was kind of fascinating too. And this is like, in the days when the satellite tags were just starting, we didn't actually have very much data from them. So it's been fun to like learn about sharks, and that there's all these different technologies that are always sort of contributing to the state of our knowledge. All right, next question. What shark species has the longest lifespan? Wait, oh, yes, Greenland shark. That is right. So we have a lot of slides, so you all should come up and claim it afterwards. So this is a Greenland shark. It looks gnarly, right? And it lives in fairly cold places. You can sort of see its distribution here, but it even comes like pretty far south on the eastern seaboard, which that was all found through satellite tracking on a Greenland shark. And this shark also, it is listed as vulnerable by the IUCN. The way that we actually know the age of a Greenland shark is through its eyeballs. And so I found this fascinating, that your eyes actually, there are additional layers of lenses that form as you get older. And this is partially what sort of contributes to like cataracts and like needing surgery as you get older. And so my student John Kuntz, they've been doing this for a while in the fish community, but he had this idea to peel the eye lenses from a leopard shark and analyze the isotopes in them. I was kind of like, all right, sure, like, let's try it out, let's go for it. So he came up with this project, and sort of the idea of this project was, how many of you have seen a leopard shark? You know, they're really cute. That's the sharks that I actually had in captivity when I was in grad school. So he caught leopard sharks from these various places. They were in the lower South Bay, Drake's Estero, which is a marine conservation area, Tamales Bay and Bodega Bay. So these locations are probably like fairly familiar to some of you. And he peeled individual layers of the eye lenses. So this is a shark eyeball, and he, like, under a microscope, is like peeling these out. And when I saw it, I was like, oh, that looks like a blooming onion. Like, if you've never eaten one of those, it just like totally like kind of pops open. You can peel the layers out and then analyze the isotopes in them, and this is what you found. So on the x-axis here for these graphs, there is age, and then on the left side, this is carbon for you, and then on the right side is nitrogen. And each location, or each region, is a different colored line, and we've sampled about 9 to 10 individuals per location. That's not a huge sample size, but when you're peeling these individual eye lenses, it's quite tedious, and so there's only so many samples that we can do. For reference, I've also given you this gray box of like what the data of particulate organic matter looks like in the central San Francisco Bay, and then coastal mussels look like. And those are from different projects. And so, sort of along what we would expect to see, that the lower South Bay sharks, these here that were caught there, they are, you know, plotting close to the central San Francisco Bay, whereas these coastal, or the ones at Drake's Estero, they're plotting with the coastal mussels. So in terms of carbon, this is doing a really good job of aligning with, you know, where they're caught. But we can also see that in this embryonic time here, where we've hypothesized is the embryonic time, that they all follow the exact same curve. So what we think is that's indicating two things. One is that the steep decline here during the embryo phase, that's because that the mom, sharks actually, a lot of times the mom has eggs like in her uterus, and so the sharks actually grow from the yolk sac that egg is producing. So they don't necessarily all lay eggs, which you may be familiar with thinking with mermaids' purses and things like that you can find on the beach. So when it's sort of gestating, that's eating that yolk sac, and that the yolk, it actually has quite a bit of lipids, fats in it, and the lipids, they have a lot more of this carbon-12 relative to carbon-13. So that part is like a physiology part, the direction of the line, but then that they all follow the same pattern sort of indicates that they're probably all sharing sort of a similar nursery area, that the moms, when they are pregnant and feeding, that they're all like in this shared area. And then they all seem to sort of also follow the same trajectory and then plateau out sort of where the sort of adult or subadult diet might be. And so we actually think that in the Central California area that there are actually these subpopulations that are forming, but that they share an area where the moms gestate, and then also where the pups are born. And we think that that area is in the San Francisco Bay here. And that really has really strong implications in terms of conservation. You think about how many people live around the San Francisco Bay, all of our wastewater, and that is getting dumped into the I know, the space, yeah, it's not good. And that is really changing the nitrogen cycle of what's going on inside of the San Francisco Bay and the ecosystems that are living there. And so then when you look here at the nitrogen graph, you can sort of see that almost all the locations are doing very similar things, except for this lower San Francisco Bay that has this really elevated nitrogen isotope value. And part of that is because like our waste is basically like the base of the food chain in the San Francisco Bay. So that we're altering sort of this baseline nitrogen isotope of what is going on. And you can see that very clearly in terms of the sharks. And one of the things that the leopard sharks are facing, but all shark populations are facing, are these dramatically declining populations. This is from a study in Queensland, Australia. But we see the same data everywhere, both in like North Carolina, and I think that we've also done this study in the Pacific as well. But this is for hammerhead sharks here. The numbers are declining. They call this one a whaler shark, and it's declining. Tiger sharks, they look like they're kind of flat, but you can sort of see a trend here in the upper numbers. And then white sharks are also declining. It's interesting to me that we have the least number of data points for white sharks, and that sort of suggests that those are harder to actually, that there are not as many of them in the waters. Sorry, I forgot to orient you to this axis here. On the x-axis is date, so it goes from 1960 to 2020, and then on the y-axis is catch per unit effort, so that we're catching fewer white sharks per unit effort. Is really signifies sort of what we've done in terms of our ocean ecosystems. And the impact of fishing is what dominates the signal throughout. And so when people have looked at like what is the biggest threat to shark populations, it's fishing, no question about it. But there's actually this other process that's happening that we are contributing to, and that's climate change. But fishing is, we're getting the largest individuals and it's really rapid, whereas climate change is much more gradual and smaller incremental change. And so that's where I'm interested in sort of thinking of looking forward. And here's another image by Ray Troll of some various sharks. And it's really hard to look at climate change, especially at the top of the food chain, because it's complicated what it does in terms of like the ecology. So we can measure things like sea surface temperature change, that's on the top figure here, where it's increasing in temperature, decreasing in temperature. This is from satellite data. We can also measure net primary productivity changes from satellites looking at chlorophyll concentrations. And then we can look at the patterns between these two, between how does temperature and net primary productivity interact with one another. And so this is this bottom figure here. And it's a checkerboard. There is no one answer in terms of what's going to happen. It is really dependent on the location and what part of the ocean that you're in. But the other thing that's notable is that we don't really have data for these mid to high latitudes. And at the high latitudes is actually where we are having the biggest magnitude of climate change. Even more than what we could have predicted, it's increasing at 2 or 3 times the rate. You've probably seen images of like polar bears not having sea ice to like hunt on and things like that. And so this is happening quite rapidly, and it would be good to sort of have an idea of like how are we going to confront this and what are we going to do about it, because we are part of the problem. So my next question is, when did sharks originate in Earth's history? Does anybody know what the oldest shark fossil is? Do you want to try? Megalodon? That is not the oldest. We will talk about megalodon at the front. Yes? Cambrian, not quite that old. Silurian, yes. So the fossil record from about 400 million years ago. So I like to think of it that sharks represent this successful evolutionary strategy that survived multiple global mass extinctions. And so here we have the geologic timescale from Ray Troll. The Silurian is here. So the oldest shark fossil we think is like 450 million years ago. Dinosaurs are during the Mesozoic, this green part. So dinosaurs came and went, and then this is the Cenozoic, and we're like way up here on the top. So dinosaurs came and went. And sharks, no problem. So this is what is called a spindle diagram. Sharks are highlighted in the blue here. Every gold star is an extinction event. And you can see some of them do not even impact sharks. Right here, the Permian-Triassic boundary, that actually impacts them. That was an oceanic event. But then the KT boundary, which is right here, that is what took out the dinosaurs. Not a blip in terms of sharks. So they are really good at what they do in the oceans. And yet, we're messing it up for them. Like, that is notable. And so this geologic record, it really offers this past climate change experiments of what's happening. And from studying that, we can look at what shapes the ecological resiliency of a species today. So what is the likelihood? What is the tolerance of a shark to be able to survive? So here along the x-axis is time in millions of years, and then the color of the bar is the climate state. And so we have warmer times called warm house and hot house in these red and oranges, and then cool house and cold house in these tealish colors. And so you can see that, you know, we've actually sort of oscillated and had these transition periods throughout Earth's history. And most of this information is actually from these things called phorians, which are protists that live in the ocean. They're tiny little shelled organisms that we get from these deep sea ocean cores. There's these huge boats that go out into the ocean. They were funded by the National Science Foundation, but the National Science Foundation now are currently being shut down to a large degree. But we would get these sediment cores and then slice them up, take out the phorians, and analyze the isotopes in them. And the isotopes tell us something about the temperature of the water, which I'll get to. So similar to how we've done this with phorians. Phorians are oftentimes from deep sea ocean sediments, but I was interested in looking at this for sharks. And so I'm really curious in what does the chemistry and the shape of a shark tooth reveal? These are all shark teeth that are found from this time period called the Miocene, which is about 10 million years ago, in Florida. And you can see that they are really different. When I started working with sharks, I thought that they were all just like pointy triangles, and it's like, oh no, there's lots of variation, you know, like there's some skinny ones, there's some thick ones, there's these with multiple cusps on them, these things with like little jagged edges, and in theory, the shape should correspond exactly to, or not exactly, but give you an idea of what the shark is doing, like something that's like really slender and pointy. You can imagine that it's like biting into like a squid or a fish. And then sharks don't actually chew their food. They just put it in their mouth and like try to digest it. And then when they have these serrations, like you think about your knife set and you use different knives for different functions in the kitchen. And so when they have serrations, that's good for sort of slicing through muscle tissue. And while we can say generalities like that, it doesn't correspond 100% to what the shark's diet is. And so that's why I was interested in sort of looking at the chemistry of the teeth with these isotopes. So now I'm going to ask the question of what is the largest shark to live on Earth?
The megalodon.
Yes, there we go. It is. And so the Meg. This is me on set here. This is a recreation of megalodon jaw. We don't actually have a full set of megalodon teeth that's been preserved fully intact. Sharks, they don't have bones. They only have cartilage, which is like what your nose or ears are made out of. And so it has a lot of organic matter, and therefore it's not as good at preserving. And sharks also, they don't have ribs or appendages like we do, and what like whales and dolphins would have. That is all cartilage that is holding them together, and they only have a vertebral column in the back. But the Meg, the teeth are big. This is someone's hand, Harry's hand, with a megalodon tooth. I have very small hands, so it does not look as impressive when I hold one. And you know, the Meg is awesome. They've even made like 2 movies about it. Not necessarily completely scientifically accurate, but you know, there might be Free Willy, but there are very few other movies, entire franchises of movies made after a single organism, and much less like a fossil extinct organism. So this is like really a shark, a fossil that has like captivated the curiosity, I think, of the general public. The other part that's good about having big teeth is that they're good for science when you're trying to get at chemistry. That's really hard to get at. So whenever I would show someone a shark tooth, a megalodon shark tooth, their question is like, what did it eat? And for a long time we didn't have answers. But we're actually getting closer to having an answer with isotopes. So our first project in this was to look at how do white shark and megalodon diets overlap. And we use zinc isotopes to look at that. Zinc isotopes, they operate a little bit differently. Zinc is a really important enzyme for a lot of cellular functions. And so zinc isotopes, as the isotope value goes down, it goes up in trophic level. So as you go higher on the food chain, you have lower zinc values. So when you look at specimens from about 20 million years ago, like in Germany, white sharks and megalodons, they look really different. Megalodon are much lower trophic level than, white sharks are lower trophic level than megalodon. And then as you, here in Malta and Italy, there's starting to be more overlap. And then as we get more recent, at 2.5 million years, there's complete overlap between megalodon and white sharks. This isn't that many samples, and this was just as we were starting to learn about this methodology. So we still have more projects going on. I have a bunch of white shark teeth. I think that they're about 15 million years old from Japan to analyze. And so to be continued in terms of this zinc isotope story. The other thing that we did was that we started to look at this thing called mineral-bound nitrogen. So all of us on our teeth, we have enamel, and when you go to the dentist, they paint fluoride onto it, and the idea is that the fluoride, it actually strengthens your teeth. Sharks already have teeth that are pre-fluorinated, because that's just naturally how they came, and that's why we have shark teeth around for so long. When that mineral matrix is forming, there's actually a protein scaffolding where it starts. And then as the mineralization, as it becomes more mineralized, that protein actually gets reabsorbed. But small bits of the protein get trapped within the mineral matrix, and that is this mineral-bound nitrogen. So my collaborators, they're at Princeton, and they were able to actually analyze these trace amounts of leftover protein crud almost, like in the enameloid. And so we measured from the megalodon to a shark that we knew was eating fish, the piscivores, and we're able to sort of look at the offset between them. And so what we found is that here is megalodon, but these are other Otodus species within the Otodus lineage. And even here, there's Otodus obliquus in the Paleocene, which would be about 66 million years ago. And what this mineral-bound nitrogen told us was that even before megalodon became really big, the Otodus lineage got to megalodon with big size, that they started being really high trophic level even before that. And that kind of makes sense that you have to be able to have the energetics and the capacity to be able to maintain your large size. And there may be other drivers like competition that are driving towards a large size. But the first part of that building block is that you have to be able to eat high on the trophic, high on the food chain before you can start actually amassing. And so this was kind of an interesting concept, and we're sort of looking further into this mineral-bound nitrogen. And for nitrogen, it's nice because we measure nitrogen in modern sharks too, so we can actually compare them. And what we found is like megalodon's like off the charts. Like it really, it did eat high on the food chain. And many times, like people think that megalodon is eating baleen whales, but if you really think about it, baleen whales are big, but they're actually not at the top of the food chain because they eat krill. And so it's kind of an interesting thought experiment in terms of like how much variation that you might see in megalodon diet if they're eating both like marine mammals like seals and sea lions, but marine mammals like baleen whales. The other thing that we see with isotopes is that they had an elevated body temperature. There's this thing called clumped isotopes where you have a carbon-13 and oxygen-18 instead of a carbon-12 and oxygen-16. And this doubly substituted combination is all thermodynamic. And with that, we were able to analyze a megalodon here. These are Mississippi whales, and then this is white sharks. And so you can see that actually the megalodon, it had a pretty high body temperature, even on par with marine mammals, maybe even a little bit warmer than what we have for white sharks. So white sharks, they can elevate their core body temperatures, and this has benefits in terms of being able to swim faster, go into deeper waters where there might be different prey, but to be warm-blooded, it actually takes a lot more energy. So you need a lot more fuel to stay at that elevated body temperature. There's a possibility that they were just so giant, like with some of the dinosaurs, we think that they were so big that their body temperature, their bodies couldn't shed the heat fast enough. So that's one possibility, and we're kind of looking into it still. But this was something that was like really interesting, because when you're that big as megalodon, then it's like, okay, like how did you maintain getting enough food to be able to keep that size? Okay, this clock just scared me. And climate change was likely what led to megalodon's demise. So this is, I showed you the figure of like how climate has been changing. This is in a little bit more detail. On the x-axis is still time. Here's sort of more historical time right here. These are climate model projections. In this area of thousands of years, this is from like looking at ice cores. So even in ice cores, you can measure the isotopes from it and see how temperature has changed. And then here, millions of years ago, this is all data that is generated from those orions I was telling you about. On the y-axis is change in temperature. This is 0 related to this average around 1950. And so this really gives you an idea of how much climate has been fluctuating. And the time that megalodon went extinct is in this purple box here. And so you can sort of see that there's this gradual change in temperature. We think that there's this really big restructuring of the ocean's circulation patterns and productivity patterns. And so if you're that big and you're warm-blooded, so you need to eat a lot, this could really, that things were changing in the oceans, could really limit sort of what your prey base was. So that's our running hypothesis right now, and we're kind of still doing more work to analyze this. The other time period, which is like my favorite, is this time called the Eocene, and that is from 56 to 34 million years ago. And this was also a huge change in climate, where we went from something called greenhouse climates. We think it was maybe like 12 to 15 degrees warmer than anything that we knew. And then it went into sort of this ice house period where we started to have glaciers forming at the North and South poles. And so the reason why I'm really interested in this, and a lot of other scientists are interested in this, is that you see where this curve is, and it sort of lines up with these worst case scenario projections from the climate models of what we could be going towards. So it's almost like an analog for what modern climate could be. And during this time, we had this fossil sand tiger shark that we have fossils from all the way from the Arctic to the Antarctic. And there's lots of teeth. Like in the Arctic Ocean, there's been something like 16,000 shark teeth found, and they're all from this one species of sand tiger sharks. And I usually get my specimens from museums. I don't actually go on expeditions. I think in my 20s it sounded very cool, but as I've gotten older and that I have 2 young kids, I think they would be very sad if I went. Actually, Miss Lisa is my kid's teacher, and she can tell you my kids would be very sad if I went for that long. But so I go to museums and I find boxes of teeth like this, and I like dig through them, and I'll measure the height of the teeth, and that's what the data that you're seeing here. So this is tooth size on the x-axis here, and these are different locations. From the Arctic, here's in dark green, Banks Island, that we have the Antarctic here in Seymour Island, and then we have these Gulf of Mexico sites here, Red Hot Truck Stop and Whiskey Bridge. And so I like measured these when I was a postdoc, and then I was like, I don't know what these actually mean. Like, I can tell that it's like big sharks and little sharks, but what is the significance of this? Unfortunately, I was able to work with a good friend of mine, Justin Yankel, that helped me sort of analyze this data. Don't remember if I put this slide in. Oh, no. And so he helped me build this model of like sharks, they grow at different rates, whether the water is cold or hot. And then, you know, that if the temperature of the water is, that there's a high seasonality, like you can imagine at high latitudes, that winter is actually still cold there, even though, and dark. And so sharks might not want to live there, they'll move. So he helped me build this model, and we found that these areas, the reason why they're colored green at the Red Hot Truck Stop in Banks Island is because they're actually nursery areas. And so you can kind of see that in the Gulf of Mexico that, yeah, the smaller sharks are here at Red Hot Truck Stop rather than Whiskey Bridge. And then the smaller teeth are here at Banks Island rather than Seymour Island. But there's a lot of overlap and the data is kind of messy. And so it's like really important to have this model to compare to the actual data that I collected for interpreting the data, or interpreting the biology. The other thing that I've started to do is, in terms of comparing models, is to compare the oxygen isotopes. The oxygen isotopes, they tell you something about the water conditions, mostly temperature, to climate model simulations. And so here on the left side is, tell them is like sort of the different layers of rock in at Seymour Island, that's in the Antarctic. And on the y-axis is oxygen isotopes, which are related to water temperature. And again, like, I got this data and I was like, okay, there's a lot of variation. There's no difference between these Helm things. Like, what do I do with this? But then it was when this climate model came out, and the climate model actually has isotope tracers inside of it. And so then I took the temperature from the climate model, the oxygen isotope value from the model, and I predicted what I would expect the oxygen isotopes of the shark teeth to be. And with this prediction, I was able to say that, okay, inside this green box is when CO2 concentration was 3 times pre-industrial levels, and right here in the blue box is where the CO2 concentration was 6 times CO2 levels. Because the world operates very differently, especially in terms of ocean circulation in these 2 states. And so the sharks, they actually overlap with that green box, which is where we also see the overlap with the invertebrates, the shells, the plants that are found in Seymour Island as well. So this is kind of cool because it's like one of the first ways that we've been able to pinpoint that, like, oh, Eocene climate, while we had high carbon dioxide levels, it was only 3 times higher rather than the 6 times higher. And so sharks existed throughout this time, and you can actually see here that there's a lot of these dots that actually go beyond those green dots. So it's like the climate model is not even predicting as much variation as what the sharks actually do. And this is just sort of a figure that we compiled, and so you can see these are these telms that I was telling you about, and then we show the clams that are there, and then the different types of shark teeth. And so in this Telm 5, there's a lot of diversity of sharks, and sharks can continue on. Some of these species continue on as we get colder waters. This is the time actually when the Drake Passage is open. So Drake Passage is the area between the Antarctic Peninsula and South America. So Drake Passage is opening, and sharks are still around, but they can't hang on at some point. They can make it to some degree, but they can't make it all the way through. And today there are no sharks in Antarctica. So the last part of my talk, I just wanted to talk about that the key to all science is actually scientists. And this is just some of the folks that have been involved in the work that I have presented to you today. On this side, on the right side, are my various graduate students doing field work. I actually do have students that do soils work, and then we have work going on with dire wolves and gray wolves as well. And then on this side is actually all of the undergraduate students that are in my lab. And so one of the reasons why I'm standing here in front of you today is because of my college education. Like, I was, my family had immigrated to the US. No one in my family had ever gone to college here before. I was like, what is the SAT? And then I had to like go home and explain it to my parents as well. And so when I showed up at college, I didn't know about these things like internships and like a job. My parents were like, no, don't get a job, just study. And it's like, well, actually, like I can get this job working in a lab, and like that's good. And so I really place a high value on getting students really engaged and involved in the research process. Jen, do you want to cover the slide, or you want to? All right, so at UC Merced, we have various majors, and here I've listed them for you and highlighted some of the new majors. But these majors here, environmental systems science and physics, we actually require research credits for completing those. And while most of the time students are like, oh man, just another thing to do, I think that's really important. And I actually do think that in classrooms, we can even give these types of experiences. So this is in my stable isotope class. We go out to the Smart Farm and out to the Vernal Pools Natural Reserve. We actually collect soil samples to sort of see how nutrient cycling is different with different land use. In my intro to marine science class, we went on a field trip to the Monterey Bay Aquarium. And having these moments of sort of like curiosity and sort of like dipping your toe into like data and research, I think is really key. And people are always like, oh yeah, like did you get lost somewhere that you're a shark scientist at UC Merced? And it's like, no, you can do research from anywhere now. It's not that you have to be doing this place-based research all the time. Like, we have airplanes. And for me, like my fossil shark teeth, people just send them to me, and I analyze them in my lab. And then I think that also key is like learning outside of the classroom. For me, some of my most influential experiences was that I did a study abroad program in Zimbabwe and South Africa. And then I also did a field study program because I was a geology major in the Western US. And so, you know, being outside, collecting data, sort of exploring and seeing how messy science is, and that the most important skill to doing science is actually problem solving. But that's not something that you can do just like, you know, when you step out of a classroom and then have problem solving. You actually have to practice that. And so, you know, going out and doing field work, being an assistant, and sort of seeing what that process is of the problem solving in real time is really key. So for us, we have things like the California Ecology and Conservation Program. We have the Yosemite Leadership Program. This helps give students experience working in Yosemite National Park. Hopefully that continues, given how funding is going. And then the students can actually get jobs as park rangers, and those are like actually really competitive jobs to get. And all the parks, they actually not only employ the folks that, you know, take your money at the toll gate and see you in the visitor center, but they employ tons of scientists as well. And so there's a park geologist that I went to grad school with at UC Merced. There's parks geologists. There's education programs specialists. We also have this program called Field Curious, where this is for folks that have never done any kind of field work or like any kind of research experience, and they get a weekend where they get to sort of try it out and see what it's like. And one of the key parts to having a good experience is actually a sense of community and a sense of belonging. And so that is something that we really focus on. And then I also have here sort of an image of the UC Education Abroad program. There's tons of programs as part of it. And you can do things like go to New Zealand and study marine science for conservation. And so for me, I actually did choose where I went to college based on the study abroad program. I really wanted to go to Zimbabwe. I thought that, I don't know, I was just like, there's not that many Z countries. Like, I could knock those out. And that was just a really transformative experience and really changed, you know, to see I did like urban and rural homestays, and life is very different in Zimbabwe than it is in suburban Atlanta, let me tell you. And so that was a really eye-opening experience for me. And then I think that lastly, then to have these authentic science experiences to help build a science identity. So I have students go to Florida and do this field school program. Two of my students are there on this boat, and they learn how to work with sharks out in the wild. My student Ashley here, she has presented about Swedish orca ecology in January at a conference in Atlanta with me. And then I think that the closing piece of being doing authentic research is actually publishing. That's like our units of measure. That's how we like give the worth of people somehow. That's weird. But here I actually have my students, my undergraduate students, highlighted in green. And so for my undergraduate students, not only do they generate the data, I ask them to plot it and to give me some interpretation and contribute to writing, and then they get to be a co-author. And so then they are, really sought after graduate students like Lauren. She's actually at UGA right now for grad visitation. Pedro, he was a PhD student at UC Santa Cruz, and Ashley is going to go on the market pretty soon. So, thanks for coming to my cool shark show. I hope that you thought that was cool, even though it's kind of warm in the room. The takeaways that I would like you to have is that diet and habitat preferences are recorded in the chemistry of our tissues. White sharks and megalodon diets really vary. Leopard sharks, they actually have these subpopulations, but they share this lower San Francisco Bay as a nursery area, and these areas really need to be protected, even though they are the most impacted by humans. Sharks are persistent, but they can be affected by environmental change, and the key to science are the scientists that are doing it. And so right now you may have heard that in the news that this is a real problem, that I think that there is an undervalue and appreciation of the scientists. There are also lots of scientists in the federal government. And so in terms, like when I talk to my undergraduate students, their number one concern is the environment. And so I really think that we need to continue giving this importance of science, the importance of conservation, because we're going to actually need people to fill all these jobs that folks are getting fired from. So that is sort of my last thing that I wanted to end on. So here are my acknowledgments on my funding agencies and collaborators that I'll leave with. Not take any questions.
But you maybe weren't ready to or didn't know how to, so did you go into the studies hoping to work on sharks, or what was your intention when you first got into this?
Yeah, so this is sort of like with the scientific method that they teach you, that you have to go in with a hypothesis and a question. And sometimes I just get really excited and I just want to generate some data. So even today, like I went into the collections with Arnold into the Great Valley Museum area, and I was like, oh, look at my isotopes on these, you know? So yeah, sometimes I just generate data for the sake of data. Like one is, like, you can always make a story out of the data. Like there's usually something there, like as long as nobody's done the exact same thing. And then the second is like, sometimes I went in and I did have a question, like with that white shark data, I did have a question. I was like, ooh, this data does not answer that question at all. Like, what do I do with it? And so I've had to be very patient sometimes and waiting for, being like, hey, like my friend Justin, I actually knew him in grad school. I was like, hey, Justin, could you build me a model and like, can we like do something with this? But that meant that study took like 5 years longer than where I intended it. Even the Antarctic shark stuff that I started, that idea started when I was a postdoc at the University of Chicago. And so that was when my first child was born, which is 2014. So that concept started in like 2012, 2013, and then just now starting to see sort of the publications that are coming out of it. So, I mean, there are people that are very fast at like generating the data and also writing it up and things, but it doesn't have to necessarily be like a race. Like, it's not always the hare that wins, right? Like, I am the tortoise. And just being sort of slow and steady and patient. And like, really, for me, it's like, I want to be a good scientist, and I want to also give opportunities to other people in terms of doing the science. And so my science is slower, but at the same time, like, I'm very proud of it, and I think that's great.
Hello. I was reading that because of global warming, the polar ice caps, of course, Arctic ice cap is thinning, and it's opening up the area for orca whales, and that orcas eat sharks. And so they're very worried about the shark population, especially the Greenland shark. So I was just wondering about that.
Yeah, there have been some really interesting behavioral changes in terms of orca. And so the last year that I was on Shark Week, we had like this whole conversation about it. And so I think that there is a little snippet of our conversation, but that was the funnest part about filming that episode, because it was myself and then 3 other actual scientists. It was not like the reality TV part that you get to see. But we were actually talking about orcas, and we were watching videos of like orca interactions. Like, orcas are going after sailboats and the Gibraltar Pass or Gibraltar Straits and stuff like that. So it very well could be that that orcas, that this opens up a lot of potential prey for them. And it probably would be devastating because orcas, they're warm-blooded, so they can withstand colder waters. Because they're warm-blooded, they have a lot more red muscle and they can swim faster. They work in teams and they can actually communicate with one another. Like, we don't actually think that that's the case for sharks. And so there is a possibility. I personally don't have problems with like changes in the food web if they're natural, but the problem here is that it's not. It's really human-induced that is instigating this change. So that was kind of a non-answer, Miss Lisa, but that's kind of what I got for you. Where's the box? Oh, yes.
So you said that you work with geology and earth sciences a lot. What inspired you to work with sharks and marine biology more?
This was when I was a grad. So I am like one of those people that like, I did not want to be a paleontologist when I grew up or a marine biologist. I wanted to be a lawyer, which is weird. But I got involved in this whole stable isotope research thing because I met a professor at like a campfire in college, and he was like, told me about stable isotopes. I was like, okay, cool, yeah, I'll do that. And then I was like, oh, you can do a lot of different things with stable isotopes. And so then when I went to grad school, my first project in grad school was looking at groundwater surface water exchange of agricultural nitrates. So like all of the fertilizers that they put on crops, and then that goes into the groundwater streams and stuff like that. That is like so far from what I do. But what I found was that when I was in grad school, I was falling asleep reading all the scientific papers, and I was like, ooh, this is gonna be like a really long time in grad school if this is how it's gonna go. And so I was in this lab though where some people studied sea otters, some people studied sloths, other people studied crocodiles. I think we had some like whales, saber-toothed cats, like all kinds of things. And I was like, oh, maybe I need to get me an animal. Like, this is pretty fun, like talking with all of my lab mates. And so I just kind of went out on a limb and I asked my professor, I was like, hey, can I analyze some sharks? And he was like, if you can figure out how to get sharks, like, sure, go for it. And I think that he really underestimated, like, what I was capable of. And so, yeah, I, like, came back and I was like, so I can go to Mexico and see all these sharks in a fishing village? Like, can I do that? And he was like, all right, sure, like, whatever. And then similarly, I was like, I think we need to do, like, a captive study on sharks and these isotopes because their physiology is really weird. It's like, if you can figure out how to get sharks onto campus and keep them in tanks, like, sure, like, I'll support you. And I was like, oh yeah, I actually know some light plumbing and construction and concrete pouring. So yeah, it was like all these like extraneous skills and just being persistent that kind of like got me through it. But it was not like necessarily me just being like, I love sharks. Like, what can I do with sharks? And that isn't necessarily where it started. Yeah.
I'm sorry if you already discussed this, but we were wondering how and why are sharks able to survive so many extinction events?
Yeah, I did not discuss this, and this is like the question that is really at the heart of like all of my research. I think it's two things. This is just me sort of giving an educated guess based on my science till now. I think one is that sharks, for the most part, are ectotherms, or cold-blooded, so they can actually go for a long time without eating. And so some of my leopard sharks, I actually had a really small sample size, and that was because some of my sharks went on hunger strike. They like did not eat for like months. And I actually have to like answer to an animal review care committee, and I was like, these sharks aren't eating, and they're like, yeah, that's not gonna work. So, so sharks can go for a long time without eating. They're cold-blooded. They also, most of their body is made of liver, and livers have a ton of fat in them. And so when sharks especially start to go into sort of like starvation mode, they can actually use the reserve energy from catabolizing their livers, like using up the energy of their livers, so burning the fat in their body to survive. So I think that that's one part of it. And then I think that the other part of it is that they are incredibly flexible. They are not picky eaters for the most part. And so, you know, there are a certain number of white sharks and also megalodon from what I've been able to see, and they have this huge flexibility of what they're going to eat. They are not my children. My children are very picky eaters. But yeah, they've really been able to manage like eating a lot of different things. Does that answer your question?
It does, thank you.
Yeah. Yes.
This is not a very scientific question, but what's your favorite shark?
Oh, it's a sand tiger shark. Somebody asks me that every single time.
Yeah.
Yeah, that's like the most asked question. So yeah, it's a great question. Sand tiger, Carcharias taurus. That's the shark that I found in the Eocene, and it's just like my model organism now. It's a mesopredator. It's not even at the top of the food chain, just kind of hanging out in the middle. I think you can throw that, but then that requires that you have to, like, that.
I don't have very good
Yeah.
aim.
Neither do I. That's why I was not offering to, like, be part of the catching or throwing.
I was wondering, do you think that there is any value in genetic research and like genome mapping of marine life in relation to studying climate change and like how, like predicting climate patterns in the future?
Yeah, I mean, I think that genome mapping is really important. Figuring out why some individuals or some populations might be resistant to, you know, I mean, even they say that sharks don't get cancer, but I don't think that's actually true. Finding out like why certain subpopulations, certain individuals are resistant to disease, like the phages of different bacteria and microorganisms in the ocean and things like that, that's important. And that's going to be really important for actually how the animals actually get through climate change, like the most acute periods of time. And it could be that in different areas that there's selection for different traits. And so ideally, we would try to have conservation across that diversity in terms of genetic variation, because that's what's going to keep them sort of resilient to things that even happen long after we're gone.
Sorry. How long does the analysis take for the shark teeth, and what does the process look like?
Yeah, for the organics, it really depends if you're looking at dental collagen, which is like the inside part, not your enamel. We have to demineralize it, so we put in HCl and react out the carbonate, CO3. with the HCl, and then that isolates the organics. And then if we think that there's like lipids, fats in it, we also use an organic solvent like petroleum ether to strip out the lipids, because the lipids will affect it. If it's muscle tissue, we actually just do the lipid removal with organic solvents, and then we actually, for shark muscle, we have to take out the urea. Sharks, they retain their urea for osmoregulation. Sharks are super weird. Like, this is how you make a whole career of something, and pick like the weirdest animal out there. Or group of animals. And then we're analyze it. And so for that analysis, you know, like, it really depends on the efficiency of how quickly people can pipette and like multitask, like multiple sets of samples going through. But then like actually the instrument analysis, it takes like about 7 minutes per sample. And so it's super short, and it's $6.50. So I was telling you about the satellite tags at like 10 or 15K a pop. And like here, it's like, yeah, like a single muscle tissue sample is $6.50. So even in my classes, I do the same with students where we like take people's fingernails, and your fingernails are also made of protein, it's keratin, and we analyze the stable isotopes in fingernails, and we're like, all right, who's actually a vegetarian, like a good vegetarian, and like who's like kind of like sneaking bacon, you know, or like how much corn is in your diet, like how much high fructose corn syrup and tortillas are you eating is also something that we can look at from the isotopes in fingernails. So that's just kind of like a little fun activity that we do. All right, I think we have. Oh, good job. Excellent.
All right. What is your favorite extinct shark and your favorite extant shark?
Oh, I think it's all sand tiger sharks. So, like, extinct shark would be the extinct variety of Stridolania macrota, and extant would be Carcharias taurus. And it's like, especially in the last, trying to think how long. The last 10 to 8 years, like, I have like really gotten a lot of mileage out of those two species. So I have to like give them some love. I don't think that other people necessarily love them that much. You can see Carcharias taurus, the modern day sand tiger, they're almost in any aquarium. They're like the ones with like the really gnarly teeth that are like poking out everywhere. That's my beloved sand tiger shark. But you also get a tote, because I remember that you answered the Silurian question.
Oh, I grabbed like a thingy already.
Oh, okay.
But actually, no, I will take the tote.
Yeah. Right for your groceries, you can save some money, 10 cents. Yeah.
Always get the free things.
Yeah. And a lot of people did not come and claim their totes. So I have extras if people want. Yeah. Yeah, there you go. All right, next question.
Hello. I
Hi.
have a weird thing. I know you're talking about how sharks are not picky eaters, and with the context of climate change and rising temperatures, I instantly think about like red tides and the more occurrences of that. So then more organisms are accumulating like biotoxins. Are we seeing that sharks are also taking in more biotoxins? Are they resistant to types of biotoxins?
I don't know if they're resistant. I do know that I have worked with Dr. Okahira. I forget his first name. He's with the California Fish and Wildlife, and then also Dr. Ree at the Monterey Bay Aquarium, and done some necropsies. And they have said that they actually look, they do histological slices of the brain, and they really look in the skull area. It's not actually the skull, but they look at that for signs of increased parasites, and then also they have some markers of damage that is caused by sort of these toxins. So that's not necessarily my area of specialty, but from sitting in these somewhat random necropsies that I've been part of, I have heard that I do think that they are susceptible. I mean, there's really a lot that's unknown about sharks, and folks have like really painted sharks as like, they don't get cancer, like there's they're indestructible, they're modern day fossils, and that's not actually true. Like the sharks that are around 400 million years ago were very different sharks than what we have today. And they've gone through like a lot of iterations of experimentation of like what worked and what didn't. Yeah, I think that there's still a lot to be learned about sharks, and as far as I know, there hasn't been a ton of research done specifically about like harmful algal blooms and sort of the linkage with shark physiology. And I think that the other part to that is like, just because, like a lot of students, they come to me and they're like, I want to study sharks. I'm like, okay, what do you want to study about them? And they're like, I don't know, like I just love sharks. But then the other part of it is that they have this really specific question and they're like, but I only want to study this in sharks. And it's like, well, actually, if you were to like study harmful algal bloom effects on like fish or like marine mammals, and then you came back to study sharks, that's actually way more helpful and you have a lot more context for interpreting the data. And so to be more flexible in sort of like what study systems that you can probe at questions at.
Hi, my name is Fred. I was wondering, do you do any research in the Bakersfield area?
Yes.
Because I was wondering, I do a lot of the, at Shark Tooth Hill.
Mm-hmm.
I've gotten some mako, great whites. I've never gotten the megalodon that they say they have there. But I was curious, they call that area a breeding ground.
Mm-hmm.
And it's no longer, it's 800 feet above sea level right now. But when you have a breeding ground like that, way back then, 1.5 million years ago, whatever they say, is that still, in your research, is that still a current breeding ground area? In that Bakersfield area?
Yeah. So I think that because I'm, maybe it's because the methodology that I use is chemistry, that I am like very quantitative about things. And so even for determining like this nursery or adult sites with those sand tiger shark locations, I actually like really wanted that model because I wanted a quantitative way to fix like what I was saying. So a lot of things that I found in the shark literature, it's sort of like someone is adding their interpretation to some qualitative data. And so for me, in terms of like, there's no way to get at like male and female sharks from their shark teeth. And so to be able to say that it's a breeding ground, I find would be really difficult for me to be able to make that next interpretive step. If there are a lot of sharks' teeth together, a lot of times what's happening is that the ocean currents are such that they're bringing all those teeth together and sort of winnowing them and concentrating them in one place. I think that there's, still like a lot of technological advances that can be made in terms of like how we interpret the data. Like even the stuff that I'm doing now, like I wouldn't have been able to do it in grad school, even though like I had these ideas in grad school, but the technology, the types of methods just weren't really around. So I'm not saying that it's impossible to be able to get there. My project in Bakersfield is actually the McKittrick tarsies and looking at the dire wolves and the various other mammals from the tarsies and comparing the ecology of the McKittrick tarsies area to the La Brea tar pits with their like dire wolves and other. That one's got like a lot more carnivores in it. So it's comparing the two sites. So totally different system.
Thank you.
Yeah. I want somebody right behind you. Wow, all of you want to still be here on Friday night. That's awesome.
If needed to, would sharks cannibalize for survival?
Oh, yeah. No, yeah, absolutely. And so this is something that I brought up at dinner, actually. So I was telling you that, like, that mom sharks, that there's the eggs inside, that once those eggs hatch and the babies start to form, sometimes not all of those babies actually get to be born because they eat their siblings. And so it's like the sharks that are like the biggest and fittest eat their siblings. and then are the ones to survive and be born.
Wow.
That's right. I know, right? Like, ooh. Yeah.
Thanks for being here tonight. It's really
Yeah.
good to listen to you. Two questions for you. Why did you choose to go work at UC Merced? And then we've got a lot of brilliant science students here. What's the reason that they should come and join you there at UC Merced?
Yeah, one of the reasons that I chose to come to UC Merced was specifically for the student population. My parents are immigrants, and I think that there needs to be equitable opportunities and access to being able to engage in science. And I don't think that the traditional ways of teaching in a classroom are necessarily the best for diverse student populations. I think that there are different cultural norms to how information is learned that suit certain folks and others. I think that these, what oftentimes are called like newfangled learning approaches, they actually help everyone. In the classroom, I don't think that it is just benefiting a certain segment of the population. I think that it like sort of affects everyone and raises everyone up together. So that is what brought me to UC Merced. I mean, there's also a job opening. So there's not a lot of job openings for what I do because actually the instrumentation is quite expensive. So there's a job opening and the student population. I'll be like totally transparent there for you. And then, what was the second question?
Why should students join you there?
Oh yeah, why should students join me? I think that of the UCs, that it is the place that students can get involved in research the easiest. The professors that come to UC Merced, they actually have opportunities to be professors at other places. Like I was a professor at the University of Kentucky before I came to UC Merced. And I'm from the South, so like the South is fine for me to some degree. That was before Trump was elected. So you know, the professors, they have other options. They're there because they want to be there. They are there because they believe in the mission of the institution and that they deeply care about teaching for the most part. It may not always show in terms of like how they deliver the material, but they do care. There are very few professors at UC Merced that I have come across that don't deeply care about giving opportunities to students. And that it's not just all about the research, you know. And when they do the research, they open their doors to students. So students can get involved, just come up and talk to a professor and be like, so are there opportunities in your lab? I have, you know, a lot of students that want to become marine scientists, marine biologists. And so I was like, hey, why don't you start a marine science club? I mean, this was actually Jen's idea, because Jen was like, you can't do this all by yourself, Sora. You're like, oh, you're right. And so like, but there's that capacity at UC Merced that students can, you know, have that leadership role and have that innovation and still chart new paths forward because we're still relatively new. So those are the reasons that I think are exciting for UC Merced. I mean, you can get a great education anywhere. You can learn a lot on YouTube at this point, but what you can't do is, you know, really get engaged with research at a really deep level. Like you need infrastructure for that. You can still come. You can. Yeah.
Maybe I misunderstood in the presentation, but on one of the graphs, I think it was like years plotted at the bottom, and then oxygen 15 plotted on the side, and it was like a temperature graph. So how do we use oxygen 15 to tell what the temperature was at different points? Like how does that work?
Glossed over that because I was like, oh man, I think I'm running short on time. But you all are the crowd that has decided to stay until like almost 9 o'clock on Friday night with me. So that is oxygen 18 versus oxygen 16. Oxygen 16 is the more common isotope. Oxygen 18 is less common. And so how oxygen isotopes are incorporated into teak mineral especially is temperature dependent. So it is dependent on the oxygen isotope of the water that it's formed in, and then the temperature determines how much oxygen 16 versus 18. So with the different temperature, there's different vibrational energy of the bonds. We're gonna get nerdy here. The different vibrational energy of the bonds, and so like whether the oxygen 18 bonds, because oxygen 18 is a little bit heavier, and so it might, with higher temperatures, it's gonna like have more energy and be excitable versus the oxygen 16. So even with like a glass of water, if you were to like leave it outside in the wintertime here, you know, because the temperature is lower, there's not going to be as much separation of this oxygen 16 versus 18 during the evaporation phase. Whereas if you were to leave water outside, you know, in the summertime, when it's really hot, there's a lot more energy in the system, and there's going to be a lot more evaporation, and so you're going to have more differentiation. And when that evaporation is happening, it's going to be that the oxygen 16 might evaporate faster because it's a little bit lighter than the oxygen 18. So all of these factors of like ocean sea level and things like that also play into the source of what this oxygen isotope pattern is based on. So it's like really, it's down to like chemistry and thermodynamics. I think that people think of me as like, I'm in a shark cage and like, I'm doing all this stuff. And it's like, no, I'm just like sitting around thinking about chemistry and thermodynamics.
My question was regarding the image of all of the shark teeth that you had and the phylogenetic tree. Could you explain how finding the stable isotopes within the tissues helps to support the phylogenetic branching that we saw?
Yeah, you know, I think that eventually it would be great to see sort of like what is the dietary variation and also the temperature tolerance of different species of sharks or different groups of sharks in relation to one another. And I would hypothesize that, you know, the more flexible you are, the more different temperatures that you can do, the more different dietary ways that you can go about, like the more successful you're going to be in terms of like your longevity in the tree of life. But that's to be determined. Yeah, like there's many more careers to be made with stabilized isotopes. Like, right, when I started my PhD, there was 4 papers with stable isotope analysis in shark tissues. And now I think that there's on the order of like 200 to 300 papers a year being published. And so I am not publishing those papers. Like I've actually kind of like bowed out of a lot of the modern shark ecology because I'm a geologist at heart. And so I do a lot more of like these geology studies. Most of the modern studies I do is with an organization called Minorities in Shark Science, trying to give opportunities to early career scientists and especially folks in the global south to be able to generate isotope data. So yeah, there's like many careers still left in sharks and stable isotopes. I think I saw that.
Last question.
Yes.
Do you think that sharks will be able to like survive the climate change since they survive like other extinctions before?
Oh yeah. Like, they're not all gonna survive, but they'll be here, you know? Like, cockroaches, they will also continue to be here. We may not be here, we definitely will not be here, but, you know, the problem is not necessarily like, whether all life is wiped out or not. It's like, we should be concerned because we're not gonna be here. Like, how much longer do we have on this planet? Organisms that are at the top of the food chain, which we are, like, we don't last long. So, that's really the concern.
If you have any other questions, please come up and ask her. Wouldn't it be great to be in our research group? It would be a blast, wouldn't it?
I have flyers here about the environmental system science. Yeah, I have flyers, so please, if you're interested in transferring. And Jen is here with me. She's the undergrad advisor. She can talk to you all about it.
Oh, good.